Hello, my name is René Ranzinger, and I'm going to introduce the GlyGen project to you. GlyGen is a database project funded by the NIH Common Funds program. This database stores information about glycans and proteins. For each type of molecule, it has a very rich set of associated data. Things like identifiers, names, species, sequences, cross-references, publications, and expression data. But for each type of molecule, it also stores specific information. For example, for glycans, motifs, classification, or related glycans, and for proteins, functions, homologs, disease, annotations, and pathway information. In addition to that, GlyGen also has the concept of sites, which are particular amino acids within the proteins that have additional data associated with them. Things like mutation or phosphorylation. All this information was retrieved from other databases that store these glycans and proteins usually separately, but integrated into one database in GlyGen, which allows us to create direct connections between glycans and proteins. For example, glycans can be associated with a particular site on a protein if that site is a glycosylation site. But there are also direct connections between glycans and proteins. For example, if the protein is an enzyme that synthesizes a glycan, or if the protein binds a glycan. All this information is available on our web portal, which you can find under GlyGenorg. To access that information, the easiest way is to either start with the Explore menu, which has different search options for glycans, proteins, or sites, or you can also access them directly via the homepage. If this got you interested, please feel free to go to GlyGenorg and explore the data. Every data is freely available under the CC by 4 license. You can also find additional YouTube videos on our YouTube channel explaining different parts of GlyGen, and we also have a Twitter account to share information.